Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 2 for May 2023. Let's begin with the first question. Question 1. The photograph shows part of a celery plant which has been placed into a beaker of stain. So this is the plant and we see the beaker containing stain. A student made a drawing of a cross-section of the bottom of the stem of the celery plant before it was placed into a beaker of stain. So we see this is the cross-section. Then they say 1 hour after being placed into the beaker of stain, the student observed a cross-section from halfway up the celery stem. Which of the following would be a different color due to the presence of the stain? So this is going to be the xylem because they have lignin, and presence of lignin is going to allow for staining to be seen clearly. So I chose Z as my answer, which is corresponding to D. The next part says explain why after 1 hour this part of the stem would be a different color. This is because the xylem transports water and mineral ions, and the stain is going to be transported away to other parts of the plant, so that place will have a different color. So I said because the stain will be transported through the stem towards other parts of the plant, the xylem transports water and mineral ions. In part B, cells in the leaf contain organelles that store molecules of starch. Name one organelle in a leaf cell that stores starch. This is the amyloplast. How many of the following show the structure of part of a starch molecule? If we look at this, we can see glucose molecules. These are all alpha glucose molecules arranged, and they have 1,4-glycosidic bonds between them. So this is showing part of an amylose. If we look at this structure here, there is rotation. This group here is down while it's up there. It's rotated in a similar way we see in cellulose, so that is not right. And when we look at this one here, we have 1,4 as well as 1,6 glycosidic bonds, so this is similar to the structure of amylopectin. There are only 2 structures that are similar to that in starch, so I chose C to be my answer. Down here they say give one role of starch molecules in a leaf cell. Starch is used as a storage molecule for glucose. In part C, transparent plastic film can be made from oil-based plastics or from starch. The photograph shows transparent biofilm made from starch. So this is the biofilm. They say give one reason why the use of starch-based transparent biofilm is more sustainable than the use of oil-based plastics. So this is because when they use starch, it's going to be a carbon-neutral process, and also starch is renewable as well as biodegradable, so that can be sustainable. So this brings us to the end of question 1. Let's continue to question 2. In question 2, eye color in humans is an example of polygenic inheritance. 2 of the genes giving rise to this characteristic are found on chromosome 15. Part A says there is more than one version of each gene involved. Which term is used for different versions of a gene? They should be an allele, and A is my answer. In part B, inheritance of different versions of these 2 genes shows genetic linkage. Explain how the phenotypes of an individual can show genetic linkage in the inheritance of 2 genes. If 2 genes are linked, it means they're positioned on the same chromosome and they are in close proximity with each other. So the different versions of the gene are closer and they're not separated during crossing over. So they're always inherited together. Therefore, the phenotype will show characteristics due to both genes. And again, remember, they are linked, so they are on the same chromosome, they're in close proximity. It's harder for them to be separated during crossing over, so they're always inherited together. In part C, one gene can give rise to more than one type of protein through post-transcriptional changes to the messenger RNA. One of these changes is removal of introns. Explain how different proteins can be synthesized from this messenger RNA that has had introns removed. Once the introns are removed, the exons can be rearranged in different orders to create different mature messenger RNAs that can be translated to produce multiple proteins. So I say the exons can be rearranged in different orders to create different mature messenger RNAs by enzymes. Some exons can be removed to create various shorter mature messenger RNAs, so the different mature messenger RNAs are translated into different polypeptides at the ribosomes. So this brings us to the end of question 2. Let's continue to question 3. Question 3. The photograph shows a caterpillar and fluid inside a pitcher plant found on the island of Madagascar. The fluid contains extracellular enzymes which are digesting the caterpillar. So this is the caterpillar inside, and then the extracellular enzymes that are carrying out digestion. Part A says describe the role of the Golgi apparatus in the formation of these extracellular enzymes. Because they are extracellular enzymes, they have to be secreted out of the cells. So the Golgi apparatus will modify the proteins, which are, of course, the enzymes. It will then package them into secretory vesicles for exocytosis to take place. In Part B, the digested insects are a source of nitrogen for the pitcher plant. Give two ways in which this source of nitrogen is used by the pitcher plant. Nitrogen is contained in many substances within a cell. One example is proteins, so the nitrogen is going to be used in the formation of amino acids, which can be used to make proteins. Also, it can be used to make nucleotides for the formation of DNA, RNA, as well as ATP. In Part C, bacterial cells have also been found in the fluid in pitcher plants, which three structures are found in both plant cells and bacterial cells. This is natural because bacterial cell walls are not made up of cellulose, they're made up of peptidoglycan. And in Part B, they say cell membrane, DNA, and pili. This is not found in plant cells. They say cell wall, DNA, and capsules. These are not found in plant cells. Here we have cell wall, DNA, and ribosomes. This is true. So the answer is going to be D. The next part says the fluid of the pitcher plant contains water and molecules from digested organisms, which bacterial cells need for growth. Explain two other conditions needed for optimum bacterial growth in the fluid of the pitcher plant. Because they said optimum bacterial growth, we have to consider temperature, we have to consider pH, we have to consider the possibility that some organisms actually need oxygen, meaning for aerobic respiration. So here I chose optimum temperature, and again here they said explain, so I have to give a detailed explanation. I say this provides appropriate kinetic energy so that the collisions between the enzyme and substrate occur at the fastest rate. And for optimum pH, the changes in pH affect the 3D structure of the enzyme, hence the shape of the active site, so optimum pH ensures a faster rate of reaction. You could also include other factors like oxygen, because some bacteria carry out aerobic respiration. So this brings us to the end of question 3. Let's continue to question 4. Question 4, the diagram shows some processes that occur after the fertilization of an egg cell. So we see fertilization, we go to the zygote, several cell divisions, we see structure A, and then cell division and specialization, we see structure B. So in Part A, they say structure B is the blastocyst. Describe the structure of a blastocyst. So here I drew how the blastocyst would be. We can see the outer cell layer, and then we see the inner cell mass. So there is also a cavity. So I said it contains an outer layer of cells. It contains an inner mass of pluripotent stem cells. Of course, these have already differentiated somehow. They're just pluripotent to form the organism. They're not going to form the tissue surrounding the placenta. It also has a hollow cavity, which is what we see here. Next, they say structure A contains 60 cells. The mitotic index of cells from structure A was calculated as 15%. Calculate the number of cells in structure A which are in mitosis. So mitotic index is cells in mitosis divided by total cells times 100. I multiplied by 100 because this was given as a percentage. And that means if this is 15%, it's going to equal to the cells in mitosis, I call that X, divided by the total cells, which they've given us as 60 times 100. Making X the subject, it gave me X is equal to 15 times 60 divided by 100, which is going to be 9. So there are 9 cells in mitosis. In Part C, structure B contains pluripotent stem cells. Histone modification and DNA methylation are processes that occur when totipotent stem cells develop into pluripotent stem cells. Explain how these processes alter activation of genes in the cells. DNA methylation adds methyl groups onto DNA, making it less available for transcription. And histone modification includes acetylation as well as methylation. In acetylation, acetyl groups are added, and then in methylation, methyl groups are added. So here I said in histone modification, methylation or acetylation of histones occurs. The DNA can therefore wrap around the histones tighter or loosely depending on which process has occurred, methylation or acetylation. Tight wrapping prevents the genes from being transcribed, so a protein will not be made, and this gene will therefore not be expressed. In DNA methylation, methyl groups are added to cytosine. This causes the genes not to be transcribed, so translation into a protein will not occur. So this brings us to the end of question 4. Let's continue to question 5. Question 5. The photograph shows a humped cichlid fish found only in Lake Tanganyika in Africa. This is the fish they're talking about. A behavior adaptation shown by the female humphead cichlid is that fertilized eggs are kept in her mouth until they hatch. Which of the following is a term used to describe the fact that this fish is found only in Lake Tanganyika? This is B. It is endemic to Lake Tanganyika. Then this is suggest why the female humphead cichlids have this behavior adaptation and give a reason for your answer. The idea of keeping the eggs in the mouth until they are hatched is number one, for protection. The eggs are protected in the mouth and they are given good conditions until they hatch. So this increases the number of offsprings that survive. So I say the mouth provides safety for the eggs and gives good hatching conditions. This should be gives or provides good hatching conditions. So more eggs hatch into cichlids. This increases the survival of the offsprings. In part B, the relationship between the length and swimming speed of sperm cells of 30 cichlid species was investigated. The graph shows the results of this investigation. So we see on the horizontal axis mean length of sperm cells, and on the vertical axis we see mean swimming speed of sperm cells. Overall, we see as the mean length increases, the mean swimming speed increases. That is a general trend I can see. So they sustain the relationship between the mean length and swimming speed of the sperm cells of these cichlid species. I say as the mean length of the sperm cells increases, the mean swimming speed of sperm cells also increases. And they say a scientist wanted to assess the strength of the relationship between these two variables. Which statistical test should be used to assess the strength of the relationship between these two variables? Because the graph shows a correlation, here I say the correlation coefficient should be used. So just one advantage for a male cichlid of producing sperm cells with a faster swimming speed. If they have a faster swimming speed, they're going to reach the eggs faster. They're going to have a higher chance of fertilization before they're destroyed. So I say the faster the speed of the sperm, the faster it reaches the eggs, the higher the chances of fertilization before being destroyed. And in part C, a sperm cell contains an acrosome, a specialized lysosome. The diagram shows a sperm cell. This is what we see here. Then they say which leopard structure is an acrosome. The acrosome is structure W, you can see there, so A was my answer. Next they say calculate the magnification of the sperm cell shown in the diagram. Give your answer to two significant figures. Here you had to get the actual length. So from my diagram, my actual length was about 39 millimeters, which is about 3.9 centimeters. So 1 millimeter is going to be 10 power 3 micrometer, meaning 39 millimeters is going to be 39 times 10 power 3 micrometer. So since magnification is image size divided by the actual size, it's going to equal to 39 times 10 power 3 divided by 60, which is what we have there. So I get 650 as my answer. Here they say, compare and contrast the structure and function of lysosome and acrosomes. In compare and contrast questions, we have to give similarities and differences, and here the question is focusing on structure as well as function. So for similarities, both contain digestive enzymes because both of them are kind of vesicles that contain the digestive enzymes, and then both are membrane-bound organelles. The differences are acrosomes contain enzymes involved in extracellular digestion only because they are released out of the sperm, while lysosomes are involved in intracellular digestion. Then the acrosomes have a curved structure. If you can go back to this, you can see it's curved, while the lysosomes are kind of spherical in structure. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Meiosis results in genetic variation in gametes. Explain how meiosis causes this genetic variation. Now, during meiosis, there are two processes. One is crossing over and the other is independent assortment. They occur in different phases of meiosis 1, and these bring about variation. So I say during meiosis, crossing over occurs in prophase 1 and independent assortment in metaphase 1. Crossing over creates different allele combinations in the chromosomes. And again, there are different allele combinations within the chromosomes, while independent assortment creates various combinations of whole chromosomes because this changes whole chromosomes or it mixes them up in order for some to end up in a different cell as the others end up in another. So this is for variation of combinations of whole chromosomes within the gametes. Next, they say give two differences in the arrangement of DNA in a cell at the beginning of interphase and at the end of prophase 1. At the beginning of interphase, this is when cell division has ended, so the chromosome should be surrounded by a nuclear membrane, but at the end of prophase 1, there is no nuclear membrane. At the end of prophase 1, you can see the chromosomes, but at the beginning of interphase, you cannot see them because they're not condensed. So I say DNA or chromosomes are condensed at the end of prophase 1, while DNA is uncoiled at the beginning of interphase, and then DNA is in the nucleus at the beginning of interphase, or I could say it's surrounded by a nuclear envelope. However, DNA is not surrounded by a nuclear envelope at the end of prophase 1. In part B, the DNA content of two cells was compared. Cell A was undergoing mitosis, and the mass of DNA in the cells in prophase was 12 picograms. Cell B was undergoing meiosis, and the mass of DNA in the cell in prophase 1 was 12 picograms. They say what would be the mass of DNA in each daughter cell produced at the end of mitosis and at the end of meiosis. During mitosis, only two cells are produced, so 12 divided by 2 gives us 6. So each daughter cell will have 6 picograms of DNA. And this cell here in prophase 1, remember in meiosis we produce 4 daughter cells, so this is going to be 12 divided by 4, giving us 3 picograms in each daughter cell. So the answer should be B, 6 with mitosis and 3 with meiosis. In part C, plants can produce seeds from either cross-fertilization or self-fertilization. Self-fertilization occurs when male nuclei from a flower fertilizes an egg cell in a flower on the same plant. Cross-fertilization occurs when male nuclei from a flower on one plant fertilizes an egg cell in a flower of a different plant of the same species. The image shows the female and male parts of a flower. So, of course, this is what we have. They say some plants have developed strategies to prevent self-fertilization. Dead plants have only male flowers or only female flowers. Some orchids release chemicals that prevent the germination of pollen produced by flowers of the same plant. And then some plants have developed strategies to produce seeds if cross-fertilization has not occurred. The fireweed plant species from North America will self-fertilize if cross-pollination has not occurred, and the unfertilized egg cells of a dandelion will develop into a seed. So they say discuss the advantages and disadvantages of strategies shown by these plants. So I began with the first one, having only male or female flowers, like in dead palm plants, eliminates self-fertilization. This is because the male and female gametes must come from different plants. Also, producing chemicals like in orchids prevents the male gametes from reaching the female gametes of the same plant. So self-fertilization cannot occur, and this will lead to greater biodiversity. And if the environmental conditions change, the offsprings will have greater chances of survival. However, if there is no other plants nearby, or if the pollinating agents are not efficient, there will be lower chance of fertilization and the numbers of the plants will decrease. Some species can go extinct. This is talking about those that cannot self-fertilize. Now, for self-fertilization, if cross-fertilization has not occurred, this increases the chances of fertilization. However, the offsprings will have lower variation. This is because both gametes are coming from the same plant, so there is a higher chance of destruction by selection pressures. This is if the environmental conditions change. Development into seeds if the egg cell is unfertilized. The egg cells produced would be different, so the resulting seed would be genetically different, leading to variation. So this brings us to the end of question 6. Let's continue to question 7. Question 7, the photograph shows a corpse lily flower, which is this one we see here. The corpse lily plant is found in rainforest in Sumatra. The flowers can be 100 centimeters in diameter and are the largest flowers found on Earth. The smallest flower on Earth is produced by Wolfia globosa. A flower of Wolfia globosa measures 50 micrometer in diameter, which is the ratio of the diameters of the flowers of that as well as that. I started off by converting 100 centimeters into micrometer. Because 1 centimeter is 10 power 4 micrometer, these 100 centimeters are going to be 100 times 10 power 4, which is basically 10 power 6 micrometer. Now I have both units in micrometers. I have 50 as well as 10 power 6 or 1 million micrometer. I can find the ratio. So the diameter ratio of this plant to that plant is going to be 1 million to 50, which is 20,000 to 1, and D is my answer. The corpse lady plant is unusual because none of its cells contain chloroplasts. Most of the corpse lady plants grow inside the tissues of a rainforest vine plant, including inside the tissues involved in translocation of organic solids. Only the corpse lily flower can be seen growing outside the vine. The corpse lily plant obtains organic solids such as sucrose from one of the tissues in the vine plant. State what is meant by the term tissue. A tissue is a group of similar cells working together to perform the same function, or you could say to perform a particular function. Next, they say suggest how the corpse lily plant obtains organic solids such as sucrose from one of the tissues in the vine plant. Because they say the corpse lily plant has its tissues directly linked to the tissues carrying out translocation, and those are going to be the phloems, so I say they obtain them from the phloem. The phloem transports organic solids up and down the plant, and part of the corpse lily plant is connected to the phloem. The lily plant tissues can absorb these compounds by diffusion down a concentration gradient, or they could absorb them by active transport against concentration gradient. In part C, the wound healing properties of an extract made from corpse lily plants were investigated. Treatments were tested on 4 groups of 6 rats with identical skin wounds. Now they're just 6 rats, so you can see the sample size could be small. The treatments were applied to the wounds twice a day. The time taken for the wounds to heal was recorded. The table shows the treatments applied to each group and the results of this investigation. So from the table, we see results when a gel is used and no extract. Then there is a gel being used and 5% extract. A gel being used with 10% extract. So we can see an increase in concentration, 0%, 5%, 10%. And then if we look at the results, as the concentration increases, the mean healing time decreases. In number 4, we see gel plus current wound treatment. The current wound treatment had the shortest time for healing to occur. However, we do not know the concentration here, so it could be 50% of the extract and it has better results, but yet this is just 10%. So based on the information given, this looks like it's the best. And if we look at this column here, we have standard deviation, meaning the experiments were repeated, a mean was calculated, and we can be able to compare the higher and the lower to the mean. If I use these values here, when I add 21.67 plus 0.48, I get 22.15, and 21.67 minus 0.48 is 21.19. So I did the same thing for all results, and I saw there was no overlap. So the first question says, explain the purpose of the gel used in these results. Now we see that for all experiments, they used a gel, meaning this is something that was consistent, and therefore it has no impact on the healing properties. So I say the extract is applied onto the gel and the gel is placed onto the wound, meaning it's just holding the extract. The gel has no active ingredient effect on the healing process, so it can be used even in experiments where the current wound treatment is used, as well as the experiments without any extract, for fair comparison. Then they say comment on the results of this investigation. Every time you see questions about commenting, these questions want you to look at the data, how efficient you think they were in carrying out this experiment, talk about the validity of the data, and make a conclusion. So I say the mean healing time was decreased by both the extract and the current wound treatments. The higher the concentration of the extract, the more the mean healing time decreased. That's true, I already talked about that. The current wound treatment was the most effective, but of course here we do not know its concentration. At least for the others, we know there is 5% and 10%. You could add something here to say, but the concentration was not known. The sample size used of only 6 rats was small. This is true as well. So we are talking about how effective they were in carrying out this experiment. You talk about the good things they did and the things that they could have improved. The sample size was only 6 rats, which was small, for conclusions on the validity of the results to be made. And then talking about the standard deviation, there is no overlap in the mean time of healing for all extracts, including the current medication, so the results show a significant difference. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Snow leopards live in mountainous regions in Asia. The photograph shows a snow leopard on a snow-covered mountain. They say give one reason for a named anatomical adaptation of this snow leopard that enables it to occupy its niche. Use the information in the photograph to support your answer. So the adaptation I chose was it has thick fur, and because it's working on ice, we can see the conditions are cold. The thick fur prevents heat loss, so this enables it to withstand the cold conditions since fur decreases heat loss. And again, this should be FUR. Fur decreases heat loss. So the other parts, you could say it has sharp teeth. You could talk about the spots that it has. You could talk about the small ears. It has small ears and these decrease heat loss. You could talk about the camouflage on the fur color and so on. In part B, snow leopards are found in several areas in Asia. The map shows the areas where snow leopards can be found. They say describe how the biodiversity of habitats in these areas could be compared. To compare the biodiversity, we have to be able to get the diversity index of each of the habitats and be able to compare them. So I said we determine the number of different species of the organisms in each habitat. This is telling us about species richness. We could determine the number of organisms per species. This is evenness. How many of each species are there? If there are more per species, then there is greater evenness, or there is species evenness. Then we can use the data to calculate the diversity index, which is the D, for each habitat, and we compare the diversity indices from the different habitats to make a fair conclusion. You could use this formula, it's in your textbook, for the diversity index calculation. The graph shows the numbers of snow leopards in the wild since 2002. They say calculate the percentage decrease in snow leopards from 2002 to 2022 and give your answer to 1 decimal place. And again, here they're looking for percentage decrease. We can see from 2002, based on the graph, it's 7,100. And here 2018 is 2,800. So the decrease is 7100 minus 2800, giving us 5900. The percentage decrease will be the decrease divided by the original times 100, giving us 83.1%. And that was my answer. Next, they say, suggest one reason for this decrease in the number of snow leopards. Because snow leopards are predators, they need prey to survive, so that is food. If they are decreasing in number, it could be that they do not have enough food, or they do not have enough mates, or the conditions are so harsh, or the habitats have decreased, and so on, or they have other predators that are destroying or killing or eating their young ones. So I chose there is decrease in food, which is prey, and there is decrease or destruction of habitats. In part D, populations of snow leopards living in different areas of Asia sometimes develop different characteristics. These populations are called subspecies. Individuals from different subspecies are able to produce fertile offsprings. Some scientists have proposed that there are 3 subspecies of snow leopards. The table shows the names and locations of the snow leopard range for the 3 subspecies. The first subspecies is northern, the other western, and the other is central, meaning they're in different areas, but they can still produce fertile offsprings. So they say the northern and central locations are separated by a desert. The width of this region is shown by the line labeled X on the map. They wanted to calculate this desert region using the scale shown on the map, and they say give your answer in standard form. Let's go back to the map. This is the map here, and this is the region labeled X. I got this distance here and matched it with a scale. If you can see, it passes to 50 and reaches 20% of the next bar, so it's about 300. So I chose 300 kilometers, or you could use an actual ruler. Your measure is going to be close to that. So length X is 300 kilometers, which is 3.0 times 10 power 2 kilometers in standard form. I have to write this here. Let me write it here, 3.0 times 10 power 2 kilometers. Explain how these subspecies of snow leopards could form. Use the information in the question to support your answer. Of course, here we have fragmentation. We have variation that could have existed among the different snow leopards. So if they were put in different habitats, the variations they had gave them selective advantages and they enabled them to survive in order to be adapted to their new habitats. That's why there are some differences between them. But because they can still produce fertile offsprings, that means there is no reproductive isolation. So I say the different parts of Asia seem fragmented, and this could have led to isolation. The different areas had different selection pressures that could have led to different ways of adaptation to survive, and thus the visible differences. This should be S, the visible differences. The leopards among the founders had different alleles, and again, these are the founders for the different habitats. When the fragmentation occurred, the founding organisms had different alleles. Mutations could have occurred and attained alleles that gave them selective advantages and were passed on to their offsprings over generations. The species were slowly changed through natural selection. Now, the leopards in different areas gained different anatomical, physiological, and behavior differences to suit their habitats due to natural selection. And since the leopards can still breed with each other to produce fertile offsprings, it means very little reproductive isolation occurred. Lastly, they say give a reason as to why these different populations are described as subspecies and not different species of snow leopards. They are not different species because they can still breed and produce fertile offsprings. So this brings us to the end of question 8 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.